Oh, sorry. Yep. Yeah, you can certainly do that. Okay, should I go ahead and share my screen? Okay, can you guys see? So, like
No,
Albert.
not
Oh,
yet. It's black.
oh yeah, sorry. So,
Okay, perfect.
thanks Alberto for the intro and for the reminder. Alberto and I are in the same, I guess, it, what do you call that? Tenure class. That's why he remembers. So, like Alberto was saying, so feel free to, ask questions. I actually can't quite see the participants, so I won't be able to see, like if you ask questions on the chat or raise your hand. So just feel free to, like interrupt me and ask the question during that particular slide. So, just to start, these are my conflicts of interest, but just to note that, both these companies are involved in cancer therapies, but I'm just including it here all the same, for perceived conflicts. So I'm going to talk about cell therapies for HIV. And I actually want to start, and this is something I actually stole from Kate Chapinelli, who's like an assistant professor at GW. To start with the team, like want to make sure there's enough time for this before everyone rushes out. And so I'd like to do this presentation in reverse, if you will, and start with like acknowledging the entire team, both mentors, my peers, and the my own mentees that have done a lot of work that propels this these projects forward. So, I don't know how, I'm going to start a little broad just so that everyone is aware. Our program is called SETI. We started here around, I guess, almost 8 years ago. It stands for the Program for Cell Enhancement and Technologies for Immunotherapy. And it's an intentional pun on SETI, which I realize now a lot of you are probably like way too young or not even born when this was first conceptualized. But it was a really big program, right? Like it's meant to really look for little green men outside our own world, and we sent this message out into the cosmos. So hopefully with the James Webb telescope out there, there's a lot more interest again in this. But we imagine, our message will be something similar, right? Like, so we'll be hopefully treating a lot of different and related diseases, because immunology does intersect a lot of different, pathophysiologies, and you can't read it, but all of these things here are different diseases, and all these are different immune cells. But the best way I think that you could think about the program, right, is it's basically cells and genes from bench to bedside. So we deal mostly with cell-based therapies, and this is the basic substrate that we all use in the program. So the different PIs and different labs in the program. And a lot of us work with the techniques available to us using gene therapies so that we could modify these cells, make them express stuff, as you'll see in portions of this talk. The good thing I think that makes the program unique and at least for us as informative as it can is there is an active bench to bedside component in it. So currently there's more than 15 phase 1 clinical trials that are ongoing, and a lot of them with collaborators in this call too, and such that like all these three processes intermingle, and then you have basically like our mini logo here, right? So SETI. SETI incidentally is Latin for whale, and that's what you have here. And so the program basically has this one universal platform, and so we have like we want to treat these solid tumors, and so there's a whale for that. There's blood cancers, and there's a separate whale for that. Brain tumors, the same thing. fungal infections like mucor, different viral infections, post-transplant, EBV, adeno, and CMV, as well as, viruses that cause cancer like HPV. And obviously, like as this talk will hopefully share with you guys is, we also have a big imprint on HIV. I won't spend too much time here because I think a lot of people in this call are familiar, right? Like the quest for a cure for HIV is obviously because it remains a global problem. In DC, in particular, right? Like it's one of those uh, places where HIV is the most prevalent here in the U.S. It has like a prevalence of around 3.2%, which in context, if you compare even with some of the African countries, right? And the reason Africa is being compared here is it's traditionally the place that we associate with really high HIV rates, right? Like it's even higher than some of these other countries here. So, and again, like the quest for a cure involves multiple things, right? Like from understanding the reservoir and developing multiple therapies. So, en route to us going to our own efforts at using cell-based immunotherapy. So I'd just like to give a very quick overview so that the students in this call are also familiar about what's going on. I would encourage you actually to go through this recent review where a lot of these figures have been taken from, right? Like which highlight the different research efforts towards a cure. So one of the efforts involves understanding the HIV reservoir more closely. And as we know more of it, we recognize that there's, many, many dimensions in the HIV reservoir. You could look at it from, like different anatomical sites, right? And you could look at it from the different cells that are involved here. For example, like the traditional, CD4 T cells, but also DCs and macrophages here. You also look at the properties of these cells that lead to, like reservoirs and how they're maintained, like the transcriptional activity, for example. And finally, you see, like the basically the location and on the genomic level where you identify genomic integration sites, how these proviruses interact with, intracellular signaling and how the cells, ultimately recognize them or how they eventually, avoid detection. One of the approaches that you hear often is like shock and kill approaches. The shock part is latency reversal. Like latency, it basically involves a cell that is almost invisible to, for the most part, in our case, the immune system, but also to intracellular defenses, right? So in this state, the virus is difficult to target. And so a lot of the shock latency reversal approaches aims to reverse this, right? So then you have upregulation of these, proteins, right? And virus production. The problem, however, is, like there is incomplete latency reversal. So it's either, like only occurring in some of the cells because there's a very narrow therapeutic window and you can't bump it up too much for fear of harming, like otherwise healthy cells. So some of the efforts involve both, increasing the potency, the distribution of these or the targeted delivery of these LRAs, and also combining these LRAs with things like immunotherapy, which our approach could also help with. The final approach I wanted to, highlight, before we talk about cell immunotherapy is gene therapy, right? So in gene therapy, there's, I guess, roughly two kinds, right? Like one is ex vivo gene therapy, where you basically take cells, right, like from the patient, modify them in the laboratory, and then infuse them back. In this case, for example, you could you could basically like treat these PBMC so that you target, like the T cell binding epitope, binding receptors of HIV so that they can go infect these cells. Or you could do in vivo gene therapy where you actually directly deliver this virus, this vector into the patient and then have that virus, like mediate the same thing. The strategies that involve or that are currently being explored in immunotherapy, like if you imagine it being administered in the context of ART, involve both, initiating ART earlier, right, so that you have, less diverse virus strains, right, like, and lower reservoirs for your immunotherapies to contend with, and you have these multiple different immune effector interventions, like combination immunotherapies that can basically then try to target all these different virus components that hopefully with this earlier art would be simpler, would be, like less diverse, would be less burdensome, right? And hopefully at some point you could interrupt art and get, no rebound of your viruses. There are several ways to use immunotherapy against HIV. And just to highlight some of these here, people have tried using, T cells which recognize HIV infected cells, and the way the immune system, through the T cell recognizes these targets is through, the processed antigen presented by these infected cells via their MHC receptors. NK cells could also be used, like some of them target on infected cells, which could represent viruses budding off. But also, as we have also recently been looking at, there are stress ligands that are expressed by these infected cells that could be targeted by our NK cells too. And then finally, you've heard of these broadly neutralizing antibodies, right? So basically, these are able to bind to these free viruses that prevent them from infecting more cells. So the way we were approaching how we would define our immunotherapy approaches, are predicated on, what we know about this small cohort of individuals who are able to maintain undetectable viral loads and also simultaneously high CD4 T cell counts in the absence of antiretroviral therapies, or these so-called elite controllers. There's around, something around like 1% of these populations. And, some of their, I guess, properties, right, like if you will, serve as great example of the immune mechanisms needed to maintain long-term viral control. So they have, for example, like a highly specific T cell response. There's polyfunctional HIV GAG specific T cells in these patients, and they have like, really well-preserved CD4 responses. Their NK cells have like a really good ability to mediate antibody-dependent cell cytotoxicity. And there's a, like a big IgG1, IgG3 antibody response, which, these two we do know mediate ADCC as well. If you look at these specific items, right, like I think the importance of an HIV T cell response has been, well documented. Like you have HIV specific T cell clones and genetically modified T cell, like optimal TCR expressing T cells showing some activity against HIV. However, like these approaches have been limited by epitope escape and the lack of T cell help that could, improve persistence. So a lot of the previous approaches utilize just the CD8 compartment, and with good reason, right? Like you are afraid that some of these CD4 cells that you might infuse might get infected as well. But there is some promising thing there. So I think this is something that we wanted to see how we could improve on. And another one is, like some of the innate immune effectors that are being stimulated by antibody engagement. And this is actually one of the major immune correlates in the, the RV1144 HIV vaccine trial, right? So they show that, FC receptor engagement by these NK cells contributed to, the response against HIV. One of the things that do limit some of these approaches is HIV itself induces NK cell dysfunction, right? So it would be ideal to not just provide, like active ADCC, but also, like NK cells to these patients that are also less dysfunctional than they are in the setting of an infection. And finally, like the administration of broadly neutralizing antibodies has, previously demonstrated significant antiviral activity. broadly neutralizing antibodies, can, as their name suggests, right, like can neutralize HIV entry into a target cell over a broad range of HIV subtypes. And their therapeutic administration has resulted in a significant, in a significant, though transient, decrease in HIV viral loads. So what we are hoping is we could combine some of these, because one of the platforms that we work with, like both the T cells and NK cells, are very amenable to modification in the laboratory, that we think we could come up with a therapeutic that could have all three components. Just to quickly segue, for those who may not be as familiar, cell-based immunotherapies, which is mostly used for cancer, right? Like is I guess one of the arms of, like the immunotherapy therapeutic revolution, if you will, right? Like some of the ones that are more commonly known are the molecular, immune therapies, which are your checkpoints, right? Like your immune modulators. What we mean when we say these cell-based immunotherapies is directly infusing these T cells and NK cells, right? Basically expanded and sometimes gene modified in the laboratory to mediate a direct effect. So you're hopefully overcoming whatever innate dysfunction is present in the patient. These therapies have shown like really good success, especially in leukemias, right? Like it's been highlighted multiple times, not just in the field, but also spilling over to the lay media, right? And I think one of the poster child, actually, poster children in this therapy, previously was one of the first earliest recipients of the CD19 CAR T cell, and her leukemia was completely cured using this platform. She got to visit the White House here, and she's now a teenager and leading her own foundation, trying to make these cell therapies better. The way most of these cell therapies work involved infusion of actual cells, right? Like, so in here to treat cancer, or, in the case that we're wanting to describe now, it to treat infectious disease. So they're either derived from the patient or from other sources, like umbilical cord blood for some, right? Like, or healthy donors that you could make banks of. And then you make these different modifications in the laboratory. So you could either select for the population that you want, you expand the relevant clones, or you even gene modify them. So you make lots of these, you do QC, etc., and then you infuse them into the patient. Their unique, their high specificity is demonstrated in this, like video here. So in the middle, for example, you see this tumor cell, right? So all the T cells are ignoring all the other cells in the periphery, and they're mediating a very clean, apoptotic cell program in this. This is a medulloblastoma. To really, like zoom in on its precise target and minimizing all these off-target effects. One thing that we are hopeful is that because these immune cells do convert into memory later on, and the goal for the field is to potentially have these cells persist and then confer protection, confer lifelong protection. So in our T cell therapy strategies, right, like there are multiple kinds where you use, for example, each T cell has their own endogenous specificities. And what we do is we try to identify, the correct antigens, and the correct MHCs, and try to expand that. And this basically uses, like we said, these endogenous T cell receptors. However, you also hear these two things that we're not going to talk about now. One is engineered T cell receptors, where you take, like whatever you identify here, and then introduce them into other cells. So you give them new still native T cell receptors, but you introduce them, and this one specific sequence. And the other is like something that has seen a lot of success in cancer, right? Like you have these chimeric antigen receptors that then target these cells depending on the surface protein of interest. So in the case of leukemias, right, like that protein is CD19. Where this is at now, we have efforts ongoing in the laboratory, which I'm happy to describe later on, but is not part of this talk, where we're targeting specific proteins that seem to be expressed by infected cells, either stress ligands or like related to the virus envelope. But mostly our work has been focused on deriving these antigen-specific T cells that naturally recognize, bits and pieces of the virus that are presented by these infected cells. Essentially, the whole process could be, summarized in this a graph here, actually, where a lot of the work does it's almost like a manufacturing pipeline, right? Like where you tweak the culture conditions so that you expand the best antigen specific T cells that you want. In this particular project that we're going to be describing here is it involves a gene modification of these antigen specific T cells, but not to confer new recognition, but to give them an additional function, as we'll describe here. So before proceeding, any questions on like the background and stuff and cell therapies? Okay. So, we want to mediate a tripartite response against HIV. And just to review, we want to essentially have, like an ability to both target, free viruses, the infected cell here, and, viruses that are, budding off and causing stress ligand expression. Each of these will be targeted by different arms of the immune response. Like we want to target budding viruses with antibodies that will neutralize them. We want to target these budding virions and these stress ligands using natural killer cells through ADCC and through their, activating receptors. And we want to target these bits and pieces of infected cells that they present through their MHC through HIV-specific T cells. And so if we modify these HIV-specific T cells to be able to secrete antibodies, so you take care of these free viruses, and then further modify the FC portion of these antibodies so that they can better engage NK cells, then you have in this one therapeutic product, the ability to mediate 3 different immune responses. And this approach is something that we're also exploring for cancer, for brain cancer specifically. And we feel it's robust enough that we could also use it for other immune cell substrates, including, for example, NK cells. This entire work has been, the result of, my first grad student, Alison Powell, who's since finished her degree in the middle of a pandemic, actually, and has since joined this thriving biotech industry community in Boston. So here's the schematic for how we make these cells, right? So we get these mononuclear cells from blood, and you basically try to enrich this, small fraction that is capable of recognizing HIV using their natural T cell responses. So then you stimulate them with antigen presenting cells, and you stimulate that subset. But then you also, like transduce with a retrovirus containing your construct, which we'll describe in a bit. And so then hopefully you'll have, clonal populations that express the endogenous T cells and are actively secreting these engineered antibodies. These antibodies, we've had different kinds here, and I'll describe the 10-10-74 experience here. Basically, it involves, like a natural antibody, and only in this case, we switched out the FC domain to an IgG3, so that the hypothesis here was it will elicit a better ADCC response for us. And above here is the diagram of the transgene in the retrovirus. So, like some of our early experiments, and this took a while because it like I said, it entailed a lot of optimizing and, differing culture conditions, varying the culture media itself too. But we arrived at a way so that we could genetically modify these T cells to secrete antibody, right? So this figure here, measures their transduction efficiency, because our gene modification, unlike a car, isn't meant to be seen on the surface. It's basically a secreted antibody. We also transduce these cells with a marker so that we know how much is being, how many of the cells are being transduced. The marker in this case is CD19, but it could essentially be any antigen that you could introduce that's normally not on your cells, right? So your CD3 T cells shouldn't be expressing CD19. And this is, like we get, a range of transduction efficiencies, but this is where the average is. The populations don't really change much, right? Like, so the proportion of CD4s and CD8s remain the same. The purity of our CD3 populations comparing non-transduced, transduced remain the same. But I think more importantly for us, we are able to detect, these antibodies in the supernatant of these T cells. So when we use this, modified platform for enhancing our HIV-specific T cells, we find, pretty much the same thing, that we could modify our HIV-specific T cells to secrete, these engineered 10-1074 antibodies. So on the left is a measure, if you will, of the antigen specificity of our T cell populations, using the secretion of interferon gamma as a marker. So if we subject our T cells, we just put them on a plate, they don't secrete interferon gamma. If you give them an irrelevant self-antigen, they don't secrete interferon gamma. But if they see, the GAG, POL, and NEF antigens that are in HIV, you do see them reacting specifically to this. And there is no significant difference whether or not the cells have been transduced or not with our antibody construct. And this is an important test, because sometimes the act of modifying these cells introduces a lot of things to, that can go wrong in TCR recognition, and we didn't want to lose this specificity. So again, you see like a transduction efficiency, a range here, which is obviously more than the untransduced, and you do see these cells secrete. Again, something that we're trying to figure out is we want to make sure that they have almost the same level of antibody secretion. So as you could appreciate here, they're still quite, we're still in a long way from that, but they do express, a range of antibodies in the supernatant.
Russell, could I interrupt you for
Yeah.
1 second on that slide if you go
Yes.
back? So is it that the transduction efficiency is always paralleled by the antibody secretion? You don't have any that may have a low transduction but are good antibody secretors?
Yeah, great question, Doug. And so we actually don't find a correlation between, so this particular dot here isn't this one. So it's not the highest transduced cells that also have the highest antibodies, and we're not quite sure why as well. So one of the things that we changed in the current version that we're testing is we introduced more powerful promoters and different secretory signals that should hopefully make it a little more stable in terms of how they secrete this. One thing that we also didn't really fully appreciate yet is how like over time this changes, and whether there's a huge burst in the beginning that diminishes over time. So yes, I guess like to answer your question, there's no correlation that we could appreciate between transduction efficiency and antibody secretion.
And then you mentioned a bit about potential changes over time. Have you had the chance to go back and repeat completely transduction in HIV-specific T cells from the same donor lot and see whether you see the same result second time around?
Yes, so not as completely yet. We have repeated it in just 2 of them, but they still maintain the specificity, a very similar CD4 CD8 ratios, and then again transduction. But so certainly not as exhaustive yet. Yeah, we yeah, sorry, Alberto.
Oh no, finish your thoughts. Sorry about that. I had a follow-up question, but finish what you were saying.
Oh no, I was just gonna say that one of the things that we think could help is if you could minimize the source of biological variability. So a lot of this, so remember, these are retrovirus transductions, right? And so this relies on the cell to be actively proliferating. We haven't, we haven't explored the lentivirus question yet, but what we did was we shifted to a different packaging, a different retroviral backbone, and a more, I guess, uniform signal of having these cells proliferate. So instead of DCs, we start using antibodies to see if we could maintain specificity while having higher proliferation rates, in the hopes of introducing either more copies of the retrovirus in them or more consistent ones. Sorry, what was your question, Alberto?
No, my question is a follow-up on that. So is the transduction efficiency similar between CD4 and CD8 T cells, that are the two cell subsets that are the majority on your products? And do you think that by the construct being in either a CD4 or a CD8, the secretion of the antibody, because obviously they may change a little bit with the promoter, can influence how good the antibody is being produced? Or maybe CD4 are better producers than actually CD8 T cells, or the opposite?
Yeah, that's a good question. Yeah, so we haven't yet. No, we do see them present at both, and the fact that we get more CD8s is an unintended consequence of the process. It's not like we're saying, oh, we want 80% CD8s. It just happens to turn out that way after the expansion. But yeah, so I think you're right. One of the things that might impact it is because, yes, like CD4s are more known as secretors. Although related to that, one thing that one of the constructs that we're trying to see is, what if we couple the secretion signal to something, say like perforin, like that'll be more present and more something that the CD8s are more used to. But we don't have answers for that yet. Thanks. Yeah, so this one, so this is basically additional documentation that the introduction of the transgene, and like the fact that we're asking these cells to make products that they aren't used to, isn't affecting their function too much. So for example, they are still able to secrete different cytokines equally as their non-transduced counterparts. If you do notice on the left though, and this is something that we are seeing a lot more, and we don't know the significance yet. While it's not statistically significant, the fold expansion for the non-transduced cells are on average higher, and we've seen this regularly enough. It's still for some reason it's still not meeting significance, but it is a trend towards that. So I think these cells, because we are asking them to do more, and we are obviously like introducing viruses to them that could activate different things in their signaling, they are being affected in terms of this I guess replicative potential. So where we're at with this is we're trying to shift this earlier in the process, where like we try to minimize all these stimulations and growing, so that they'll be I guess, for lack of a better word, younger, in the sense that they're earlier in their expansion process, and hopefully minimize this this fold expansion difference here.
So, Russell,
yeah,
I have a question. What happened to the expansion? Instead of doing a transduction, you add soluble antibody?
So if we add, so I guess the equivalent is, so we added antibodies bound to beads, and it's an early experiment, and it seems to work much better. So we tried the Dyna beads instead of stimulating with DCs, and then
no.
Sorry.
I'm referring to 1074, to the antibody. So the cells are transduced, are producing the antibody. Is the effect on the proliferation due to the transduction, or due to the presence of an antibody in the culture?
Oh yeah, that's a good question. No, so we no, that's a good question. We haven't looked into that. No.
I mean, just in case the antibody is having some
Yeah, that's
clear
true.
effect on the proliferation of the subset. And then the second, besides the antibody, is there anything else that is being expressed from the construct? I completely missed that.
So we try to be as oh, you mean like the construct itself? No. So in theory, it should just be the antibody and the the marker, which
Okay.
should hopefully be innocuous. We cut off the signaling parts of the marker. But yeah, and then like a lot of these search for cytokines is we're trying to see if are we inadvertently causing more things, but it doesn't look like that's the case. Obviously, it's not super exhaustive. It's just a few of these, but so that's a question we wanted to ask too. Yeah, no, that's a good point. Let me see if we could look at that.
I mean, I don't know if antibodies have a role on, like in general, right? I'm thinking about the antibody in general having some influence on the expansion of CD4 and CD8 T cells. That is
Yeah, no, that's a good point. I mean, I guess in theory, I wouldn't think so, but we haven't actually tested it. So these cells do they don't express the normal antibody receptors. Still even after this modification, but again, we don't know if there might be other effects, because for sure, for example, not in vitro, but how when immune complexes form in some of the autoimmune diseases, or in some of the like the immune pathologies, they do impact the function of CD4s and CD8s. But obviously it's I guess it's a little more complicated because they see that in vivo, so maybe other cells are involved, etc. No, but that's a great question. So when we look at the antibodies themselves too, we want to see if they basically like they maintain their natural abilities, right? Like, because one of the questions has always been, well, like T cells, fine, they could potentially make proteins too, but the antibodies are involved in binding, so there might be like changes in shape, right? Like, or changes in glycosylation that would impact their ability to bind. And so what we try to do is we used a model of this cell line HeLa that expresses HIV envelope, and wanted to see if our antibody products are able to bind to these cells. So we cultured these HIV envelope expressing or non-expressing HeLa cells, and then tried to see if there are anti-IgG3 on their surface, right? So so here I'm showing an example of how some of these look like unstained, so you don't see any binding. And then if you co-culture these, if you put these HeLa cells in supernatant from untransduced cells, you don't really see any binding. You don't really see antibodies on the surface, but in the transduced supernatants, you could detect that these antibodies are capable of binding. And this is summarized, like these three different supernatants here. They're all the same, model like HeLa, and we'll try to look for additional models, but these are at least from three different donors. And I think more importantly for us is if we and we'll talk a little more about this in a bit, is if we then use these supernatants and try to co-culture these targets with NK cells, you do see like an increase in killing of these NK cells in the transduced supernatants compared to non-transduced. That is similar to the wild type antibody. So this is just the antibody itself that's not secreted by the T cells, and so just showing that this does perform similarly to the wild type. If you notice, the y-axis here is change in killing, because as we've come to see as well, these infected cells and in general the presence of envelope on the surface does impact the expression of these activating ligands, which we hope to exploit as well. So finally, we wanted to mix all these cells together into one, one in vitro experiment of anti-HIV function. And so what we put together was, like these CD4 T cells, so these are all from the same donor, right? They get infected by HIV, and then they get treated by these HIV specific T cells, some secreting antibodies, some not. And then after 5 days, we assess the level of HIV, P24, and measure, like, is there any decrease in the presence of these effectors? So here, looking first at, these T cells by themselves. So in one of the donors, for example, you have, if there's no HIV, then we don't see any HIV after 5 days. If you have the culture condition where it's just the CD4 T cells, you do see this, like really high HIV infection level as shown by P24 in the supernatant. If you mix them with autologous non-transduced HIV specific T cells, you do see a drop in a further drop when these are the transduced counterparts. And you pretty much see, like the same pattern in 3 out of 4. In this particular donor, for example, there wasn't any effect. And this is more pronounced actually when the HIV specific T cells don't have a high enough specificity. But overall, you do see, on average that there is this pattern of decrease between the two. This one because of the few numbers and this one donor not expressing any significance. This isn't significant to each other. If we do add NK cells, you do see a further decrease, and I'm going to magnify this part here. Again, you do see, there seems to be the least amount in cells that have, like NK cells, in the presence of antibody. But again, this doesn't seem to fall to the level of significance. And a large part of this is because when you zoom into the killing by the NK cells themselves, there is already a high level of killing actually. Like you have ranging from 40% to almost 80% in the absence of antibody. So there is already something in the infected cell that is causing these NK cells to attack them. We're looking actively into some of these different activating ligands, like other groups too, and they do show these NKG2D ligand expression on the surface. So we don't know when the best time for it will be, and that's something that we're looking into. And I guess, I apologize if this will happen a little faster. I'm happy to discuss it in more detail later, but just cognizant of the times, I'm going to go through the NK cell therapy, and then we could talk a little more. So like we said, one of the questions that we found is the NK cells themselves, they seem to be, having a really good antiviral response against these infected targets already, outside, these antibody expression by the T cells. So we asked then, what if we use these NK cells directly instead, right? Like, so we will take these NK cells, activate them so that they express a lot of these activating receptor ligands. And some of these activating receptors, and because some of their activating receptor ligands, for example, MIC-A and MIC-B, seem to be highly expressed in these HIV specific T cells. And then you could still combine them with, with antibodies, because they do signal through their CD16 receptors as well. And then hopefully if they are from the same donor, then inhibitory responses would be almost zero. So we basically took the same thing, and this is being prepared as another manuscript. The previous T cell work, were derived from a paper that was published in 2020. This one is, we're finishing up, but basically, showing the same thing, right? So we could transduce these NK cells, right? And then they could be secreting antibodies as well. And we wanted to, quantify the percentage of antibodies. So this is, normalized per 1 million, transduced NK cells. And then if you look at the, percentage of the NK cells that are active that actively have these antibodies in them, so using an intracellular stain, it's actually only a small percentage, like around 20%, even though the transduction efficiency is a little higher than that, right? So that's something that points to the disconnect that we were talking about earlier, that the transduction efficiency doesn't, again, we don't know why, it doesn't equate to the level of secretion. So maybe some of the cells are too tired, or they then don't get enough raw materials for
So Russell, if I could ask, and I may have missed this, did you talk about NK cells and T cells from the same individuals? Do they have the same, does a transduced NK cell from one donor have?
No, that's a good question, Doug. So I'll have to double check, but I'm pretty sure no, like that these are separate sets of donors. One of the limitations we have, and I think we'll try to get like bigger donor pools, is once we start using the cells up, it's pretty much limited to that experiment because of what we want to see after. But yeah, no, so that'll be a good point, whether it's an individual, like a per person thing. Yeah, no, yeah, I'm almost sure that these are not the same individuals from that we tried before. So I guess similar to the T cells, right? Like again, we looked at a bunch of different panels, and again, because both the activating receptors and all these other receptors are important for NK cell function, we tried to see if they are maintained, and for the most part, it looks like they are. When we also tested for cytokine secretion, there doesn't seem to be any difference so far. And just to highlight, this is not quite exhaustive yet, so a lot of this work is still ongoing. They don't lose their cytotoxic ability. This one is not an HIV target; it's just a general, K562 target. One of the things that we did want to ask our NK cells to do is, in the presence of the antibody, is if they could activate, right? And we tried to measure this in 3 different ways. One is literature has shown that CD16 starts down regulating once you mix NK cells with the antibodies. And you do see upregulation of CD107 that also corresponds with increase in interferon gamma production. And for the most part, this is what we see. So in the presence of increasing antibodies, so you have 100 nanograms to 10 micrograms, you do see decreased, shedding of CD16. The same for, production of interferon gamma here. And the cytotoxic degranulation is measured by CD107 expression. So again, showing that the presence of transgene, or the process of modifying these NK cells, don't impact their ability to engage the FC receptors. And then finally, and again, this is just 2 donors, but we do see, similar to the T cell studies, that if you co-culture them with infected cells, you do see a big drop already with the non-transduced NK cells, and then you further increase this when these cells are making the antibody. And then again, same for this other donor here. And so if you normalize these 2 donors, we have this really good pattern here. But just to again emphasize, this is still early, and that we have to do this in more repeats. So to summarize, the work so far, we've seen that both HIV specific T cells and NK cells can be combined with broadly neutralized antibody approaches in a single platform. They could secrete antibody from these non-canonical cells that don't impair the antibody binding and don't impair the ability of these antibodies to engage effectors, more specifically NK cells for ADCC. So what's going on now in Dr. Jones's lab at Cornell is, we have different groups of mice that receive basically, like transduced versus untransduced versus, transduced with NK, the same in vitro experiments that we showed you guys, and then comparing, like the ability of these xenograft mouse models to control HIV. So hopefully more to come after, I guess like a few more weeks. These mice have just engrafted last December, and so we'll see how they fare. So I guess we have a few minutes for discussion, but before that, I'd just like to thank the team. Again, a lot of this work has been in the result of Allison's really like productive several years with us. And also acknowledge a lot of our collaborators from the HIV field. A lot of this work we have absolutely zero expertise here, and so I'd like to highlight like a lot of these people who have worked with us for some of these assays. Alberto and Doug are here, and obviously like my mentor Kath and the rest of the SETI team, who's been very supportive of all our efforts. So, thank you, and yeah, hopefully we have like a couple of minutes for questions and comments.
Good job, Russell. Is there any questions from the audience?
So Russell, in terms of the in vivo longevity of these transduced cells versus other cells, have you seen any difference in preliminary data so far?
So none. No, we don't have the preliminary data yet. But yeah, so we don't know yet. Yeah, I think like we mentioned, we're doing some, I guess it's not as relevant with the cancer setting. So we'll see what it looks like on those xenograft models.
And are you also looking for anti-idiotype antibodies?
So that's a good question. So right now, no, and we, yeah, no. So do you think with the mouse model that we have it, that could be a source of poor persistence and engraftment, and if they start developing it's against the product, you mean, right? Like
Correct.
Yeah, no, that's a good question. And we've so right now we don't have any plans yet, but we are collecting like the serum for all these in several time points, and that's something that we could definitely see or try to see. It would probably be the easiest would probably just to look at just general anti-human, because these are human antibodies, because there would be no other source anyway for them.
These are the NSG, right? These don't have B cells?
No, they don't. So what we're trying to see is if there will be, so I guess not in this particular experiment. We were sort of hoping that in future versions we could try to recreate one lineage versus another, like how I forgot what is being added, but CD40 ligand, I think, right? Like where you try to encourage some of these CD34 engrafted mice with a specific lineage. But yeah, no, that's a good point.
So in the transduced NK cells, I mean, you basically like, it kills the virus. I mean, there is no like almost no P24 release, right? In the
So, in just with the non-transduced, you mean, right? Yeah, and I think. Sorry, what?
No, that with the transduced secreted antibodies, there was a pretty clear reduction on P24.
Yeah.
Do you think that everything is due to killing?
So that's a good point. So whether it's because they're being neutralized essentially. So we don't know. I guess one of the one experiment we could do is if we could try to use an alternate, like yeah, it'll have to be well controlled, but for example like the NK92s, right, that don't have ADCC in theory, and you know, again, I know it's allogeneic and will need all those corresponding controls, but that might be informative in terms of teasing out what percentage is due to neutralization, because if it is a significant chunk, then even those cells without CD16 expression should be able to, you know, show the same drop, and if not, then they should maintain. Yeah.
Yeah, it's really cool how it's working. Is there any other questions? It's 3 o'clock and you have another
yes.
meeting. Yeah, I don't want to take your time. So if there is no any further question, or if you have, you can always email Russell. And there is some questions from Katie Angels in the
Oh, sorry.
chat about.
Hi, sorry. I was just wondering if there were any publications about translating this to brain cancer. It's really a persuasive approach, I would think.
So Katie, yeah, I'd love to talk to you more about this offline actually. So the short answer is yes, and we're actually proposing an R01 on it, and we do have prelim data on some of the brain cancer models, but yeah, yes, we are very much interested in moving it to cancer, and yeah, funnily enough, specifically brain cancer.
Okay, yeah, great. Thanks. Yeah. I'll shoot you an email.
Great, thank you.
So you have some
Alberto. So just to ask, and I apologize, I even have to ask this. So some of the data are unpublished. So this is not going public, right? Because, you know, just to make sure.
So we don't have to, Kara, just be sure that
it's all internal, right?
Because normally now we post all these talks on the CFAR website, but we don't have to if you are not, if you don't want to. I mean, it's so Kara, so we'll need to be sure that is not posted on the website.
Right, that's fine. We don't have to post it.
And once you publish, Russell, if you don't mind, we'll save it. And once you publish, we can always post it back.
Yeah, no, that'll be great.
Yeah, because we have really good access on a lot of our seminars from YouTube, and people will go because the timing sometimes is not the perfect timing for everybody.
Yeah.
I do wanted to say that you have kudos for Tom, that is a community member. He liked really much your presentation and like it's on the website. So yes, that it was really accessible. So that's you know, I just want to bring it up. And
thanks, Tom.
And if there is no more questions, I will adjourn this talk. Thank you so much, Russell. It was a really excellent talk. We learned a lot, and happy that it's moving along and moving forward. And I'm going to class.
Okay.
So
thank you.
Thank you so much, Dr. Cruz.
Okay, thanks, Karen.